Alright, I'm going to make a quick little video so you guys can learn the graphic organizer for the cell cycle and kind of go through it real quick so you can kind of get the idea of it. So you've got your graphic organizer made, and you write on your first flap G1, and underneath the G1 flap you would write that it is growth. And then underneath that flap, you would write that it is normal life functions. The cell spends most of its life in this phase. If you want, you can even write that some cells never leave this phase. You don't have to write this part if you don't want to, but that is true that some cells, like some of your nervous system cells, maybe your liver cells, even some of your muscle cells, spend their entire life in this phase and they never ever leave. And some cells reproduce and go through this, leave this phase very quickly. So it depends on the type of cell. Then after G1, you go through the next phase, and then after that it is the S phase. So the next flap, you would write S on that. And the S phase stands for synthesis. So underneath here you would write synthesis. And synthesis stands for, well, synthesis means to make. And what are we making? We are making DNA. So we would write DNA is replicated or copied. So that says copied. Replicated means to make a replica or to copy. We're copying the DNA. Then on our next flap, it is G2. So G, this is the second stage of G. G again, what did G mean? G meant growth. So it is G again, growth, only more growth. And we're also going to prepare for mitosis. So when we say preparation for mitosis, what do we mean? Well, we're doubling the organelles. The cell has to make sure that it has all the organelles it needs to split in half. It has to have at least half of its organelles on each side of the cell. So it's got enough for both halves of the cell when it splits in half. Both halves of them. Again, you don't have to write what's in the parentheses. I just want to make sure you understand. So basically it's just doubling its organelles. And this graphic organizer is made really rough, just a quickie, so that you can understand while I'm not here. I just kind of roughly made it. The next stage is M. M stands for mitosis. I couldn't write the mitosis in underneath the flap because I wanted to write the stages of mitosis in on the flap, or in under the flap. So the stages of mitosis, and I don't know if you can really see them, so you might have to just have someone make sure you can tell what they say. But they are each a phase, so they each end in the word phase, and they are prophase, so pro, P-R-O, phase, metaphase, M-E-T-A, metaphase, anaphase, as in A-N-A, phase, and telophase, which is T-E-L-O, phase. And so those are the 4 phases of mitosis. And we've got some steps that are involved in each of those. There's a whole list of steps that are going on during those 4 phases. So there's quite a few things going on that we have to make sure that you get information occurring during those 4 phases. So we've got prophase, where the chromatids, now a chromatid is a single strand of DNA. So when the DNA goes to, after it's replicated, each of those strands are going to coil up real tight. So those single strands coil up real tight. So I say coil up here. And then they're going to match up with their sister. So a sister is an identical strand of DNA. So they match up with their identical sister copy, and then they form X's. So they come together and form X's with their identical copy. And those X's are called chromosomes. So here we have where it says form X's called chromosomes. And that's what happens during prophase. And then the nucleus disappears. So literally the nucleus just disintegrates. So normally you see the nucleus around the outside of the chromosomes, the DNAs, in the middle, and the nucleus is around the outside, and that nucleus just disintegrates, it just disappears. So the chromosomes are still there and they're X's, but the nucleus just disappears around it. Metaphase is a quick, easy step. Those chromosomes that are X's in the center, they literally just line up down the center of the cell, or the equator. So the chromosomes line up in the center. And then anaphase, since those chromosomes are lined up down across the center of the cell, just lined up in a straight line on the center of the cell, now those chromosomes just split. So each of the chromosomes just split, and they go to the poles. So each of those sister chromatids just split apart. And so in a line, each of them just split apart and go to the opposite poles. All in a line, each of them go apart. And then in the final stage, telophase, the chromatids, because remember, once they're not chromosomes anymore, when they're separate, those chromatids, being separate now, called chromatids, they reach the opposite poles. And then nuclei, so they're separate, a nuclei now reappears around each set. So once the chromatids have separated and gotten to each of the poles, gotten to each of the ends of the opposite sides, then a nuclei reappears around each side. And so there's chromatids around each side, and a nucleus reappears around them. And so now there's still one cell. They haven't separated into two cells yet, but there's chromatids that are exactly identical around in each side, and a nucleus reappears around. And so it's almost like it's two cells, but it's really just one cell still, with identical sets of chromatids on each end. And then cytokinesis is the final stage. So here we have cytokinesis, which cyto means cell, and kinesis means splitting. So cytokinesis means cell splitting. That's when this one cell, which on each end has these two nuclei in it, the cell's gonna begin to split. So the cell's gonna literally start to just pinch in the middle, and it's gonna pinch and pinch and pinch and pinch and pinch in until literally the cell splits into two. So it already has a nuclei in each side, and so as it begins to pinch, 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 pinch in, it splits apart, and now there's two separate cells. So once, in the very beginning, you had one cell. As this whole process begins to happen, it just pinches into two, and you have two cells that are separate, and they are identical to the original cell. That is the cell cycle.